All right, last asynchronous concept clarification number 1. We're going to talk about the basics of translocation into the ER. And we're only going to get through the little first part of it, but I do want to introduce this because we're going to talk about this and some key concepts of this also together in concept clarification number 2 in the synchronous section. So just like we introduced nuclear import a little bit, and we'll talk in more detail about that mechanism, we're going to introduce ER translocation and talk in more detail about that mechanism as well during the synchronous session. So first I have a few questions for you to answer. So the inside of the ER is the same environment, mostly the same environment, as the what? Well, the inside of the ER is the same environment as the outside of the cell. So remember that the inside of the ER basically is going to be where all those proteins are located that will then go to vesicles, onto the Golgi, and then through vesicles onto any other parts of the endomembrane system, or be secreted. So if you're a protein inside the Golgi vesicle, when that fuses with the membrane for secretion, the inside of that vesicle ends up basically being the same as the outside of the cell. So just like these proteins are released through the exocytosis pathway, if these proteins are meant to fold and function out here, they need to fold and function inside the Golgi and the ER as well. So the inside of the ER and the inside of the Golgi are relatively the same environment as the outside of the cell, which is one of the reasons they're distinct from the cytoplasm, separated by their own membrane system. So what proteins need to be targeted to the ER first? Well, anything in the endomembrane system needs to be targeted to the ER first. So if it's an ER resident protein, it will be targeted to ER first. It will have the ER signal sequence and then an ER retention sequence. If it's in the Golgi, it'll get to the ER first and then sent to the Golgi. If it's a lysosome, go the ER Golgi lysosome. Secreted or membrane protein, ER Golgi secretion. Even the peroxisomes and so forth. So anything in that endomembrane system has to be targeted to the ER first. And then finally, the ER itself, right, the ER over here, has a smooth area, smooth ER, and rough ER. What is the purpose of both of those? Well, the rough ER is rough because it has the ribosomes on it, and the ribosomes bound to the ER are actually co-translationally translocating the proteins into the ER. So while those ribosomes are making the protein, they're basically pushing it into the ER. And that's what makes the rough ER look rough, because they are dotted with those ribosomes making the ER proteins into the ER. And then those proteins eventually will be sorted and sent wherever they need to go. The smooth ER, on the other hand, does not have the ribosomes. That's not part of protein synthesis. That's more of lipid synthesis, detoxification of a lot of different chemicals, and things like that. So smooth lipid synthesis, rough protein synthesis. All right, so remember the ER has co-translational translocation. So we've talked about the chloroplast, mitochondria, and nucleus so far. That is post-translational. In other words, the ribosome will translate the mRNA and make the whole protein, and then that whole protein will be translocated into its destination. For the nucleus, it's the folded protein that goes through the nuclear pore complex. For mitochondria and chloroplasts, it's the unfolded protein that's kept unfolded by heat shock proteins gets in. On the other hand, for ER, it's co-translational translocation. Right? So the ribosome will be translating the protein in the cytosol, and then it will pause, and we'll talk more about this in a bit. It will pause, and then it becomes located to the ER, and it makes the rest of the protein in the ER. And this is why the rough ER looks rough, because it's dotted with these ribosomes that are making the proteins into the ER. Co-translational translocation. All right, so my next question for you is, what are the advantages and disadvantages of each? So what do you think the advantages of post-translational translocation is? What's the advantages of co-translational translocation? Well, one advantage of co-translational translocation is you don't have to spend energy and work hard to keep the protein unfolded. So the mitochondrial protein, remember, the heat shock proteins, the HSP70, had to keep this unfolded so that it fit through the translocation machinery and so that it didn't do the right function in the wrong place. with co-translational translocation, you basically are pausing translation and putting the protein directly in, so you don't have to keep it unfolded along the way. However, you do need machinery that's going to recognize this signal sequence, that's going to pause this, and that's going to bring the ribosome to the ER for this translocation. So this is a little bit more complex and requires some more machinery. This usually, unless it's the nucleus because of the large nuclear pore complexes, this usually requires you to prevent the protein from being folded before it gets to the right place. Alright, so let's take a look at this animation that talks about translocation into the ER.
The endoplasmic reticulum, or ER, is the most extensive membrane system in eukaryotic cells. Proteins transported to the Golgi apparatus, endosomes, lysosomes, and the cell surface all must first enter the ER from the cytosol. As an mRNA molecule is translated into a protein, many ribosomes bind to it, forming a polyribosome. There are two separate populations of polyribosomes in the cytosol that share the same pool of ribosomal subunits. Free ribosomes are unattached to any membrane. Membrane-bound ribosomes become riveted to the ER membrane and translate proteins that are translocated into the ER. These membrane-bound ribosomes coat the surface of the ER, creating regions called rough endoplasmic reticulum.
So this is important to think about. One thing here is that we have the polyribosome, so as soon as a ribosome binds to the 5' end and basically moves like a train along and starts translating that protein, when it's out of the way, another one will bind. And this is true for both cytosolic proteins and those proteins that will be co-translationally translocated into the ER. And this is also why you basically have a whole bunch of these ribosomes bound together, because they actually might be translating the same mRNA. And so this mRNA essentially is going to snake its way this way, and then this guy will be done, and then snake its way this way as this one continues, and so forth until all the ribosomes on it are done. But even as this one pulls out, two free ribosomes that are just floating around in the cytosol might bind there, and there eventually will be brought to the ER as well, as this one mRNA is used to make many copies of that protein.
Two kinds of proteins are moved from the cytosol to the ER. Water-soluble proteins completely cross the ER membrane and are released into the lumen, while transmembrane proteins only partially cross the ER and become embedded in the membrane. All these proteins are directed to the ER by a signal sequence of small hydrophobic amino acids. The signal sequence is guided to the ER membrane with a signal recognition particle, or SRP, which binds the ER signal sequence in the new protein as it emerges from the ribosome. Protein synthesis then slows down until the SRP ribosome complex binds to an SRP receptor in the ER membrane.
So this is how that ribosome becomes bound to the membrane when it knows it has a ER protein, or a protein with an ER signal sequence. So they mentioned that this ER signal sequence is hydrophobic. So when that emerges from the ribosome, that is highly energetically unfavorable. It's a long hydrophobic region, and it's in the aqueous solution of the cytoplasm. That's not favorable. The SRP signal recognition peptide signal recognition particle recognizes the signal sequence peptide sequence, and it's going to bind to it because they both kind of have that hydrophobic face, so they're much more energetically favorable for them to be bound together. And then this SRP will pause the ribosome until it brings it to the SRP receptor, which is on the ER membrane, and then that will bring this to cause co-translationally translocation, basically making the protein inside the ER as it goes.
The SRP is then released, passing the ribosome to a protein translocation channel in the ER membrane. Thus, the SRP and SRP receptor function as molecular matchmakers, connecting ribosomes that are synthesizing proteins containing ER signal sequences to available ER translocation channels. In addition to directing proteins to the ER, the signal sequence functions to open the translocation channel. The signal peptide remains bound to the channel, while the rest of the protein chain is threaded through the membrane as a large loop.
It also makes sense why this signal peptide, this signal sequence here, stays bound because remember it's hydrophobic, so it doesn't want to go on in. It's going to remain bound there, and the protein is going to basically be pushed through by the ribosome as it's translated.
Once the protein has passed through the membrane, it is released into the ER lumen. After the signal sequence has been cleaved off by a signal peptidase located on the luminal side of the ER membrane. The signal peptide is then released from the translocation channel into the membrane and rapidly degraded. It is thought that a protein serving as a plug then binds from the ER lumen to close the inactive channel. But not all proteins that enter the ER are released into the ER lumen. Some remain embedded in the ER membrane as...
All right, so this is where we're going to start. We're going to talk more about the embedded membrane proteins in class together in the synchronous section. But I'm going to pause that here and just kind of further talk about the translocation into the ER. All right, so a few things that you need to know. So we have the ribosome. The ribosome is translating the protein, and out pops a signal sequence. That signal sequence is hydrophobic, and so therefore it binds nicely to this cleft on the signal recognition particle, SRP. That signal recognition particle, when it binds to the signal peptide here, it causes a conformation change. And that conformation change basically causes this huge bend in this hinge, and that causes this domain to flop over and basically block the mRNA part of the ribosome. So it basically prevents the ribosome from moving any further along that mRNA while this SRP is bound. So that huge conformation change is really, really important. This is one of those post-translational regulation mechanisms. So when it's bound to the signal peptide, that causes the conformational change, and now it pauses the ribosome so the ribosome doesn't continue making that protein until it gets to the ER. This is a visual of the SRP. So the SRP is actually one of those proteins that is made of protein complexes and RNA. So we have protein domains, a lot of alpha helices in this binding pocket here. That's a hydrophobic binding pocket for the signal peptide. And this hinge is made of RNA, and this binding causes this hinge to basically swing, and that's what's going to pause the ribosome. All right, so after that ribosome is paused, this SRP will bind to the SRP receptor, which is on the ER membrane. This conformational change is also important to make this binding work. When the SRP is unbound or is not bound to the signal peptide in the ribosome, this conformation of it does not bind to the receptor. And that's good, because if it did, you would have a whole bunch of SRPs just bound to the receptors without anything to bring to it. But when the SRP binds to the signal peptide, causes conformation change, and blocks the ribosome from translating further, this conformation now binds nicely to this receptor, therefore bringing the ribosome to the ER. And then, as the video said, once that happens, it brings it really close to a translocator, and we'll learn about this. This is called SEC61, SEC61. And so that then opens up, and now the ribosome is sitting here, basically inserting the protein into the ER as it's translated. So my question for you now is, what three things does the SRP protein have to bind to? And I call it a protein, but remember it has some RNA in it, but I'm going to, just for ease's sake, call it a protein. But what three things does the SRP have to bind to in order to work? Well, it must bind to the signal peptide, the signal sequence. It then has to bind to the ribosome for pausing it, and it has to bind to the receptor. It does not bind to the SEC61 translocation complex, because if it did, it would block that, but by binding to its SRP receptor, it brings the ribosome to this location, and now it can translocate through. All right, so here's our ribosome sitting on the SEC61 translocator. It kind of looks like a ribosome that's basically pooping the protein right into the ER. It's sitting there, your mRNA comes across here, and the protein polypeptide will be being made here and basically just inserted right into the ER. I know that visual is pretty gross, but that's what I think of, so now you have to think about it too. The ribosome is sitting on the SEC61 toilet, pooping the protein right into the ER. Another key concept is that the ribosomes that translate our ER proteins with the ER signal sequence, they're no different from the other ribosomes. We have these ribosomal subunits. They basically are waiting for an mRNA to become free so that they can bind to the 5' end and start translating. When they bind, they are going to start translating that protein, and if no signal sequence pops out, they just keep translating that protein in the cytosol. There's your protein being made in the cytosol. Remember, that's the default if there's no ER signal sequence. But if they happen to randomly bind to an mRNA that has a signal sequence, that's when SRP does its job, pauses this, and they are brought to the ER, and now those ribosomes are on the ER as part of the rough ER. But as soon as they're done, they will fall off again, become part of the cytoplasmic pool, and depending on which mRNA they get, they will either make a cytoplasmic protein or an ER protein again. All right, the Sec61 has to open two ways. One, it obviously opens to allow the protein translocation to occur. So the ribosome would be sitting here as it translocates this protein, but they've removed that image just for ease of sake. But it would be being made, pooped into, if you will, into the ER. But that signal sequence basically has to be cleaved. So not only does the Sec61 open up to allow the protein to go into the ER, it also opens sideways to release this signal sequence after it's cleaved. This signal sequence, if it's part of a soluble protein that will be secreted or is inside the lumen of the ER, Golgi or lysosome, it's going to get cleaved off for the same reason that the mitochondrial signal sequence gets cleaved off. It's this large hydrophobic domain that can really alter the fold of this soluble protein. So it gets cleaved off once it's done its job. It doesn't need to stay on because this ER protein will never leave the endomembrane system. So it doesn't have to get back in. So that Sec61 opens sideways to release the hydrophobic signal sequence in the membrane, the nonpolar membrane of the ER, and eventually that will become degraded. But this sideways opening is really important as well for transmembrane sequences of these proteins. So when we talk in class about how the transmembrane protein gets threaded into the ER, this is going to be very important. So remember that Sec61 opens towards the ER and opens sideways to release things into the ER phospholipid bilayer as well. So that's where I'll leave this here, and we will talk about both nuclear translocation and the details of membrane proteins and how they are stitched into the ER together on Monday. See you then.